Welcome, everyone, and thank you for joining us for a presentation titled Impact of Segmentation Quality on Assay Endpoints, Deep Learning for High Content Screening. It's my pleasure to introduce you to Fuhui Long. Dr. Fuhui Long is a staff analytics engineer at Cytiva, a global provider of technologies and services that advance and accelerate the development and manufacture of therapeutics. Her work over the years has mainly focused on developing machine learning, deep learning, and image analysis algorithms and software that help address problems in the biomedical applications in life sciences. For a complete biography of our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them through the Q&A box. Our speaker will address those questions following the end of the presentation. Please join me now in welcoming Dr. Long. I will now turn the presentation over to her. Welcome, Dr. Long.
Hello, everyone. Welcome. My name is Fuhui Long. I am a staff analytics engineer from Cytiva, Issaquah in Big Seattle area. For those of you who are not familiar with us, Cytiva provides technologies and services to our customers to help fundamental biological research to develop vaccines, drugs, and novel cell and gene therapies. We were formerly part of GE Healthcare Life Sciences and joined Danaher early this year. Today, I would like to talk about the impact of segmentation quality on assay endpoints in high content screening. This is the agenda and learning objectives of today. First, I will give an overview of the high content screening workflow. Then I will give a high-level introduction on machine learning, including deep learning methods. After that, I will show how machine learning and deep learning can help high content screening. Then I will focus on the critical step for the single cell level high content screening, which is the segmentation step. And finally, I will use several case studies to show how machine learning can really make a difference. Small molecule drug discovery can be divided into two categories, target-based and phenotypic methods. Target-based methods start with a defined target and identify compounds in biochemical assays. Because the exact target of small molecule is known, lead optimization through the rational design of molecules is feasible. However, off-target effects in follow-up studies can greatly impact their desired efficacy and add adverse toxicity. Besides, target-based approach assume only one target causes a disease phenotype and ignores the complexity of biological system. In contrast, phenotypic methods start with phenotypic perturbation of cell-based systems and identify compounds in phenotypic assays. These assays are used for high content screening to identify compounds in a more physiological setting. It typically generates large amounts of data that require much more sophisticated analytics, including various machine learning methods. Today, we will just focus on high content phenotypic screening. Image-based high content screening workflow starts from microscopy imaging. The acquired image are then processed by image analytics. Image segmentation extracts individual cells, nuclei, membranes, and other cellular structures. Feature extraction then extracts various features, including intensity, morphological, and other statistical features within individual cells or cell populations to form a high-dimensional feature vector. The downstream analysis then converts the high-dimensional feature vectors into interpretable and actionable conclusions. High content screening started back in late 1990s. In 1996, Terry Dunley and Lindsay Taylor formed a company called Cellomics. They filed a patent on image-based high content screening and got approved in 1999. The invention screens a large number of compounds using fluorescent labeling and microscopy imaging. This approach, however, is based on nuclear translocation. That is, the activated cytoplasm proteins are transported into the cell nucleus, where it can initiate transcription of a particular gene or genes. This figure illustrates how to determine the transcription factor distribution in nuclei and the cytoplasm region using morphological operations. Compared to what we can achieve today, this is actually low content. Nowadays, we can do much better, obtaining thousands of features in a single cell. For example, in this study, the cell painting assay developed by Broad Institute used 6 fluorescent stains to reveal 8 cellular components in a single microscopy-based assay. Automated image analysis pipeline was able to produce about 1,500 morphological features from each cell. The protocol was later independently adopted at Recursion Pharmaceutical on different disease models. Today, assay complexity is continually increasing and outgrowing the capabilities of the current analytical methods. More sophisticated machine learning methods are under development and will bring high content screening to a new phase. Before I show you some examples of using machine learning approaches to help high content screening, let me give you a very high level overview of machine learning techniques. Today, we talk a lot about artificial intelligence, machine learning, and deep learning. Let's clarify these concepts first. Artificial intelligence is a very broad area. It applies to any technique that enables computers to mimic human behavior. Machine learning is a subset of artificial intelligence that enables machines to improve with experiences. And deep learning is a subset of machine learning that uses multi-layer neural networks. There are different types of learning: supervised, unsupervised, and semi-supervised. Supervised learning needs data annotation. In the example on the left, you need to provide the information that these 4 red samples belong to one class and the green samples belong to another. Supervised learning needs a training and test steps. Typical examples are regression and classification tasks. Unsupervised learning do not need annotated data. In the example on the right, you do not need to tell which sample belongs to which class. It does not need the training phase. Typical examples are clustering and unsupervised dimensionality reduction. Semi-supervised learning sits in between. It uses both labeled and unlabeled data. It also involves training and test steps, but has a lower requirement on the amount of annotated data and its quality. Many different machine learning methods can be used in high content screening. For example, the regression methods, including linear and logistic regression. The instance-based methods, such as support vector machine, k-nearest neighbor. Ensemble methods like boosting and random forest. Clustering methods like the widely used k-means and hierarchical clustering. Dimensionality reduction methods, including principal component analysis and linear discriminant analysis. And there are also Bayes models and many more. Recently, deep learning neural networks have been used widely in biomedical image analysis. For example, convolutional neural networks have been used in cell segmentation. Recurrent neural networks, especially long short-term memory networks, have been used in cell tracking. Generative adversarial neural networks have been used in synthesizing training data to expand the small training set, and autoencoder has been used to extract features. Now let me give you some concrete examples of using machine learning, especially deep learning, in high content screening. In typical image-based high-content screening, machine learning is playing critical roles in every step. Once images have been acquired, we can build a classifier to do quality control. If the images have low signal-to-noise ratio, low contrast, or limited resolution, we can use machine learning and deep learning methods to enhance images and achieve super resolution. To segment cells, nuclei, cell membranes, and other structures, we can use computer vision algorithms like region growing, watershed, etc. for simple problems. For challenging problems, we can use deep learning neural network, especially convolutional neural networks, to achieve very high accuracy in cell segmentation. Once individual cells are segmented, we can use computer vision and machine learning approaches to extract morphology, intensity, and texture features. We can use dimensionality reduction methods to map a high-dimensional feature vector to a lower dimensional space. We can then use clustering and classification analysis to find interesting patterns that are biologically significant. In the following slides, I will give you some concrete examples. Low signal-to-noise ratio images present challenges to the downstream image analysis. To get a high SNR image, you may increase laser power or exposure time. However, this will damage the samples, limits the possible duration of recording, and cause artifacts and photo damages. This example shows how deep learning can improve signal-to-noise ratio computationally. In this study, a group from Max Planck Cell Biology used pairwise low and high SNR images acquired under low and high laser power, trained a convolutional neural network called UNet to produce high SNR image from a low SNR image. The bottom left image shows the low SNR image acquired under low laser power. It serves as the input to the trained neural network. The second image from the left is the network output. The third one is the high SNR image acquired under high laser power. It's the ground truth. And the last one on the right is the restored image using traditional denoising method called non-local means denoising. This example shows that machine learning methods, especially deep learning method, can produce high SNR images without increasing laser power or exposure time. Deep learning has also been used in whole image classification. For example, Human Protein Atlas project aims to map the subcellular distribution of the human proteome with confocal microscopy. The current version of the database compresses image data for nearly 13,000 proteins. Each image in this dataset consists of 4 channels, the antibody stand protein of interest, and 3 reference channels to outline the cell. The human cell compresses many compartments, here defined by 28 labels. The goal is to build classifiers to predict the location pattern of the protein of interest. This dataset is challenging to analyze automatically because of the prevalent multi-label classification and high imbalance of the 28 different protein localization classes. To find the best solution for this problem, a Kaggle competition was held. It attracted more than 2,000 teams. Participants used deep learning approaches and applied various strategies, including modifying neural network architectures, designing effective loss functions, augmenting data, and using pre-trained models. The winning models far outperformed previous efforts at multi-label classification of protein localization pattern by nearly 20%. The previous example is the whole image classification, in which structural segmentation is not needed. Let me give you another example on high content screening with segmentation. This is a screenshot of our high content screening software called INCarta. It has a rich body of analytics modules. The structural segmentation module uses machine learning-based annotation, UNet with ResNet backbone, and transfer learning to segment individual structures. The phenotypes module extract single cell-based features and classify cells into different phenotypes with the combined use of unsupervised, supervised, and active learning. Here are some further details on phenotypes. After structure segmentation using our deep learning module, it extracts high-dimensional features. It then uses unsupervised clustering method to assign individual cells to different classes, which serves as our initial training set and initial classification model. Then, through active learning, users may iteratively refine the training set and train the model by correcting prediction errors. In this example, machine learning provides an unbiased overview of potentially interesting subpopulations. It provides intuitive user experience and more sensitivities to resolve subtle phenotypic differences. From the above examples, you can see that deep learning accommodates the added complexity in high content screening. The downside is that in order for deep learning to work well, one needs to have a large amount of high quality annotated data. Recently, there is an increasing amount of research on how to apply deep learning using weakly annotated small data set, which is a more advanced topic and beyond the discussion of today. So what approaches should one choose? Should we always use deep learning? The answer depends on the balance between the cost and reward. For small data set, traditional machine learning is comparable to deep learning, or even better. As the scale of the data set increases, deep learning outperforms traditional methods and can produce highly accurate results. Overall, traditional machine learning can take good use of small data set, has a lower requirement on hardware. For example, no high-end GPU or even no GPU is needed, therefore leads to a lower cost. It also has a higher interpretability. We know which features are important for a task and understand why is that. However, traditional methods produce less accurate results. They need handcrafted features and parameter tuning. They also have lower generality. On the other hand, deep learning methods can produce highly accurate results. There's no need to do handcrafted feature engineering or parameter tuning, and the trained models can be generalized much better. However, deep learning relies on big and high quality annotated data. They also have a higher requirement on hardware like GPU, clusters, and cloud setting. Therefore, they tend to have a higher cost. Besides, they have a lower interpretability. So the right strategy is to understand your problem well and choose the right approach. Having introduced machine learning, especially deep learning's roles in high content screening in general, now let's focus on a specific topic, segmentation. Why is segmentation important? Segmentation is the first step in extracting quantitative information from individual structures. Errors in segmentation will propagate in the downstream analysis. At some point, meaningful biological results can be overshadowed by artifacts generated by segmentation. Segmentation is also very challenging. This is because structures are complex and objects are crowded. Images often suffer from low signal to noise, low contrast, and are resolution limited. There is high phenotypic variability generated from natural population heterogeneity and perturbation effects. Traditional segmentation approaches have many limitations. You need to manually define what features matter. You need to define feature extraction rules. Algorithms optimized for one structure may not work for another. And you need to fine-tune your parameters because they vary from experiment to experiment, from dataset to dataset. As a result, traditional methods have limited accuracy, robustness, speed, and usability. The solution is to use machine learning and deep learning techniques to develop trainable segmentation. For example, the 2018 Data Science Bowl challenged participants to segment nuclei in a variety of 2D light microscopy images without the need for any manual interaction or adjustment of parameters. This computation attracted nearly 4,000 teams across the world. Top participants succeeded in this task, developed deep learning-based models using ResNet backboned UNet and mask RCNN to achieve highly accurate nuclear segmentation. The image here shows examples of segmentation results from the top 3 participants. The first column is the original image. The second to the fourth are the results of the top 3 participants. And the last column was produced by Cell Profiler as a reference. The segmentation maps show pixel-wise alignments between the ground truth and the predicted segmentation. White color codes correct answers, blue codes false negative, and red codes false positive. Without manual interaction or parameter tuning at all, these solutions provide more accurate results than traditional approaches. The most widely used deep learning network architectures for cellular segmentation are UNet and mask RCNN. UNet is a U-shaped convolutional neural network. It has an encoding path, a decoding path, and skip connections. It has been successfully used in many biomedical image analysis applications. Using ResNet backbone and transfer learning, the network is able to extract more generic and meaningful features that are critical for challenging segmentation problems. Mask RCNN is another type of convolutional neural network. It can achieve object detection, segmentation, and classification all at the same time. It has also been widely used in segmenting structures from biological images. Using the ResNet backbone UNet architecture and transfer learning, we developed a deep learning segmentation module called Synap for our INCarta high contents screening software in order to segment cellular structures from light microscopy images. Synap works with any type of data, including bright field images. It is highly accurate with both low signal to noise and high density images. It copes with high phenotypic variability within a field of view and across all plates in an assay. It's easy to learn and use without needing an image analysis or programming background. INCarta Synap has pre-trained segmentation models that deal with common applications and can be easily tuned to work with specific biology problems. Here is an example of cell segmentation on the BBBC dataset using INCarta Synap. It successfully segments each individual cells and did a much better job than the traditional methods we tried. Here are some further examples on cell segmentation. Again, they're more accurate compared to some traditional methods, including watershed and random forest we tried. There's no need to do any handcrafted feature engineering and no need to do algorithm parameter tuning. To test the generality of Synap, we further segmented the time series like microscopy images of wound healing assay of kidney epithelial cells. Taking the pre-trained cell model, we added a small amount of wound healing assay images and fine-tuned the model. Due to some technical issues, I'm not able to play the video. Here are some snapshots of the segmentation at different time points. The neural network model precisely separates the wound region from the remaining tissue. Here is an example of using Synap to segment cells in a video sequence from the cell tracking challenge. Again, started from our cell-based model, we fine-tuned the model using a subset of the training data from the cell tracking training set. When applied to new images, our model gave us very precise segmentation results in tracking these cells. This example shows a more challenging scenario on vacuole detection achieved by our customer, Grace Pohan, at the University of California at San Francisco, using our Synap model. Like most biologists, she realized the potential benefits of using quantitative image analysis, specifically a high-content screening assay that can quantify the amount of vacuole formation to infer the effect of a genetic modification or chemical perturbation. However, her efforts were paused when realizing she didn't have a way to quantify what she could see by eye. The image on the right shows vacuole segmentation is actually a very challenging problem. The wide variety of vacuole sizes, intensity differences, and weak edge information all make it very challenging for conventional image processing methods. To make it even worse, the nuclei exclude fluorescent label and look almost identical to the vacuoles. By using only her understanding of the biology, Grace started from our pre-trained cell model, annotated a small set of images, trained her own model, and had a working solution in a very short time. With INCarta's Synap, she's now able to get results that even an expert image analysis algorithm developer would struggle to achieve. As you can see from this image, Synap is highly sensitive without over-detecting noise or structural similar appearance. It is fair to say that deep learning is able to achieve the best segmentation performance for high-content screening applications. Deep learning methods are more reliable, flexible, and accessible than traditional methods. There is no compromising or biasing of parameters to one treatment or phenotype. It maintains high accuracy across samples with high phenotypic variability. A single workflow can deal with many different applications. Our deep learning tool can be used by a novice with no image analysis experience. Trained model can be shared and continually improved as they are trained on more data. Finally, let me give you several examples to show the roles of machine learning on high-content screening endpoints. Remember this image-based high-content screening workflow I showed at the very beginning. Image segmentation is a necessary step to enable single-cell level phenotype analysis, but segmentation is not the endpoint. It's reasonable to question if segmentation quality really impacts the final result. Errors in segmentation are often ignored as long as the errors are not visually alarming. Indeed, for simple, robust assays where high-quality control are available and biological changes are obvious, segmentation errors may have little impact. However, as we mentioned before, errors at this early stage of high-content analysis can propagate throughout the rest of the analysis and can effectively reduce the ability to resolve subtle changes. At some point, these errors add up until you're unable to detect or accurately characterize the statistical relevant phenotypic differences present in the images. Here is an example showing that accurate spot detection is critical in determining the agonist effect. In this example, changes in the number of spots counted per cell indicate a drug's ability to rescue an induced phenotype in a dose-dependent manner. We use both deep learning and conventional segmentation method, which is watershed plus multi-scale top hat, to detect the spots in assay with and without agonist. As you can see from the upper right image, conventional method detected false positives in treated samples, whereas the deep learning method produced the correct segmentation, as shown in the bottom right image. Note that traditional spot detection algorithm requires you to optimize analysis parameters on one extreme condition, test on the other extreme, and then iteratively tweak the parameters in this fashion until a compromise is reached between conditions. The bar graph on the right shows the z-values based on our deep learning model and the traditional segmentation results. Z-prime is a statistical measure of how much two distributions overlap. In this case, it measures the distribution in number of spots per cell across all cells for the two control groups. The traditional spot segmentation approach produced a much lower z-prime value compared to our deep learning model, meaning that the segmentation errors in traditional methods lead to a situation where you cannot confidently determine the true magnitude of the agonist effect. This example shows that segmentation really plays a critical role to the endpoint. The second case study will show you accurate segmentation promotes precise quantification of subpopulations in the downstream analysis. Researchers is studying C. albicans, which is a type of fungus in our GI tract. Certain environmental conditions will cause the yeast cell type to transit to the infectious rod-like structures called hypha and pseudohypha. This study looks only at the negative and positive controls in replicate. The negative control is grown under normal conditions where it is expected that most of the cells will be a yeast-like state. A positive control is grown under abnormal conditions that stimulate the transition to the elongated morphologies. The goal of the experiment is to evaluate if automated imaging and image analysis can clearly differentiate between the control populations by segmenting individual cells and accurately classifying them as one of the phenotypic subpopulations. We segmented the images using a traditional watershed-based approach and our deep learning Synap tool. As shown in the upper left image, traditional method suffers from under and over segmentation, and sometimes it completely misses the real signal, whereas our deep learning model produced high accurate segmentation results as shown in the upper right image. In the downstream analysis, Phenoglyphs was trained to classify individual cells from both segmentation experiments using the same set of extracted features. Colors in these two segmented images represent the class label, where purple codes pseudohypha and yellow codes hypha. The Synap model results agreed much better with the expected outcome, as you can see from the bar plots in the bottom right, where the negative control is dominated by the yeast phenotype, and the positive control is enriched with the elongated forms. In comparison, the traditional method deviates from the expected outcome, as shown by the bar plots in the bottom left, where negative control is dominated by pseudohypha. All the three types of errors in the traditional segmentation method can account for over or under counting objects and can confuse the classifier. The classifier is most negatively impacted by intersegmentation, where multiple cells or out of focus signal is merged into one object. The last example I'm going to show you is the systemic sclerosis high content screening that was did by our customer, a company called Labroots. Fibroblast to myofibroblast transition, in short, FMT, is a biological process in which fibroblasts differentiate into myofibroblasts and secrete excessive quantities of extracellular matrix proteins. It has been reported to play an important role in late-stage systemic sclerosis. Galapagos is using high-content screening to find novel drug targets that rescue the FMT phenotype in primary diseased human skin fibroblasts. They developed a screening assay. The readout allows them to evaluate ECM protein production in response to cRNA perturbation. The assay screening plate layout uses multiple replicates for controls throughout the plate to observe the plate and positional effects. They first used intensity-related measures. This did not yield a very strong pattern for the control wells versus the samples and negative controls, as shown in the upper right heat map. Using our machine learning tool Phenoglyphs, they developed a classifier that can successfully differentiate cell types and showed a strong separation between the controls, the negative controls, and the samples, as you can see from the upper left heat map. The figure on the bottom left shows the heat calling using phenotypic model produced by Phenoglyphs and intensity scoring model. Both approaches have a hit rate of 1.5%, but only 20% of the targets overlap. The bar graph on the bottom right shows the Z' score of assay plates. Results obtained from Phenoglyphs was far more robust with higher Z' score than the intensity scoring method. The machine learning-based approach represents more relevant targets than those identified with intensity-based approach. This improvement in obtaining relevant targets will result in fewer false positives and allows identification of targets that would have been dismissed as negative by the traditional approach. To summarize, machine learning, especially deep learning, are able to accommodate the increasing complexity in high content screening and are providing powerful analytics tools for drug discovery. Segmentation accuracy in many cases can generate significant impact to the high content screening endpoints. Finally, here is a list of upcoming webinars. The next one will be on October 26th. David Andrews from Sunnybrook Institute will introduce film fret techniques. There will be 2 more webinars in November, one in December and one in January. Please save the dates. With that, I would like to thank you all for your attention. We will be happy to take your questions. Thank you.
Thank you, Dr. Long, for that outstanding presentation. We will now move into the live Q&A portion of this presentation. As a reminder, please submit your questions via the Q&A box. Welcome, Dr. Long, and I also want to welcome Will Marshall, who will be joining us for the Q&A. Welcome, Will. All right, now we're going to get here. So, there we go. Welcome, sir. How are you doing? Thank you for joining us. How are you doing? Good. Thank you for joining us for the Q&A. And we'll wait. Dr. Long, we still don't see you and cannot hear you, but while we're waiting for Dr. Long to join us, Will, could you answer this audience member's question? What is the best use of DL with regard to scale of data? And the audience member asks, includes hashtag plates.
Yeah, so I think I would need a little more details to fully answer the question. So if you're learning, if I assume the question is if you're trying to basically learn the features that best describe a phenotype, then more data is going to be better. But within the context, I think, of Fuhui's presentation, if it's just about training the segmentation model using kind of the transfer learning approach that she described, then it kind of really, you can get by with a much less data. We're not talking on the order of plates. We're talking on the order of 5, 6 fields of view to train a segmentation model. Yeah, but it's all quite relative. And in our experience, I think the answer is you kind of have to do the experiment, because we're always kind of surprised by how we're not the best judge of how complex a problem might be. We kind of have to do the experiment and find it out later.
Thank you so much. And I think this next question is also for you, Will. What quality controls are used to rank the results from one application to another? Some may be obvious to others, very subtle. How is this measured?
What quality controls are used to rank the results? Yeah, I'd have to ask for some follow-up clarification. I'm not really sure what results and what the rank would be. So is that assay robustness, or yeah, I'd have to ask for some more clarity.
Okay. And then I know we are still waiting for Dr. Long to come back in. Perhaps, Will, you can help us with this question as well. How many, as well as what analysis is done to show for correctedness?
Yeah, so again, I need a little clarity, but correctness, maybe model accuracy. So if we're talking about segmentation, to kind of assess the accuracy of a segmentation model, it's probably best for maybe Fuhui to take this one. I'm pretty sure that she would tell you there is no agreed upon best method to do this. But things like mean IOU or something like that, to really look at accuracy down almost at the pixel level, is one metric that people commonly use, although it definitely is not the best metric in all cases. It kind of depends on what your goal is.
Thank you so much. And Dr. Long, I see that we see you now. Welcome back. We appreciate you logging out and logging back in. Can you hear us now?
Yeah, I can hear you. Can you hear me?
Wonderful. I can. Before I get to a question for you, will the audience member that wrote the previous question just clarify for us what quality controls are used to rank the results from one application to another? And the question was really about confidence of the results. Could you give us a little more detail on that with that clarification?
Yeah, so regarding the quality control, so one way is to visually check, of course. We have biologists here that work with us closely to check the results. And so they can tell us whether the results make sense or not. We have almost every experiment that we try the traditional segmentation method and deep learning. And we have biologists to tell us that which one makes more sense. That's one way. And the second way is that we have some quantitative matrix, for example, the mean IOU at the pixel level, so that you can compare two segmentations, which is more close to the ground truth.
Thank you so much, Dr. Long. This next question says, and I want to thank again our audience members for these great questions coming in. Dr. Long, is the transfer learning part of an entire CNN module, or is it on a per application or training data set?
Yeah, that's a very good question. So transfer learning is actually embedded in our pre-trained model. Our pre-trained model started from some initial weights, and those initial weights are not did not start from the random assignment. It actually started from the pre-trained weights derived from ImageNet dataset. So those are natural images. We use those weights to start from, and then trained on like nuclei, cell, and neurite dataset. And then once we train the model, we provide our customers the base model for those structures. So the information regarding very generic visual features is already embedded in the ImageNet dataset. And then the specific features of the cells, nuclei, and membranes are embedded in our base models. So those are all shipped to our customers, and our customer can use those base model to fine-tune on their own data. So when we say transfer learning, actually it is carrying all the generic information and the basic information of cells, nuclei, and other structures to our customers, and they can fine-tune it.
Thank you so much, Dr. Long. And does one always have to have a positive or negative to do the training? Because sometimes one doesn't know what the phenotype will actually be. Can you tell us a little more about that?
Yeah, so Will, you can, I just first explain a little bit, and Will can add on. So our phenoglyphs actually did not start from controlled, so it did not start from the point that you have to know positive or negative, or the different classes you should assign the cells to. Actually started from clustering method, which is an unsupervised method. Once using the clustering method, it divided data into different classes. And then start from there, we will introduce the supervised and active learning. So the answer is no, you don't have to. Will, do you want to add something on top of that?
Sure. Yeah, if the question is really specific to like a classifier like phenoglyphs, at least the way we designed and built the workflow, like Fuhui said, you don't have to start with controls. And it's important to say that it really is a classifier. So you don't get some kind of phenotypic score relative to a control back from it, although you definitely could. So the idea is that you use some unsupervised clustering just to kind of get a sense of how the data is structured, and you kind of decide in the supervised learning piece how you want to divide that up. But one thing that you can do, and it works quite well, is when you run the clustering, you only feed in the positive and negative control data, because what we find is with the supervised learning, the human's not the best at kind of being consistent in differentiating between very subtle differences in phenotypes. So oftentimes that can lead to a model that just doesn't really train well and come together because of the inconsistencies by the human. So one way to get around that is to just put in your positive and negative control into the clustering. Usually those are going to be well separated, because they're controls, and build your model off that, then apply it to the rest of the data set. So you don't have to, but there are definitely reasons why it helps.
Thank you both so much. We have time for a couple of more questions. Our next question, so what is the readout of the DL that was described? A list, a PCA, a UMAP, and with replicates, how does one assess the reproducibility of the results?
Okay.
Sorry, Will.
Go ahead, Fuhui. I'm just saying that was a complex question, and I'm not seeing it in the Q&A. Could you repeat it, maybe?
Sure. So, what is the readout of the DL that was described? That's the first part. And a list, a PCA, a UMAP, and with replicates, how does one assess the reproducibility of these results?
Yeah, the output of the deep learning is the segmented results. So if you feed the deep learning module, the Synap and image, then the output is the segmented results. If you repeat multiple times, once the model is trained, if you repeat multiple times, the result is there. It's not changing each time. So there's no randomness inside. The randomness only introduced when you do the training. So yeah, it's repeatable, and the output is the segmented images. I hope this addressed the question.
Thank you so much. And did you want to add to that, Will?
Okay, yeah. So I see. So yeah. So Fuhui is correct. If we're talking about the deep learning that was mainly focused on here, it would be a segmentation, but I think a segmentation map. But if the question was kind of talking about an approach where you're learning the features, then it would make sense, right? Would we use PCA, UMAP, TISNE, or something potentially? If that's a question, I don't know. Maybe this isn't on the question, but one thing that kind of interesting is run all of your images through a network like the painter's network that learns kind of just really perceived features. Use something UMAP or TISNE to just plot all the individual fields of view, so you can visualize them and see how they cluster together. And yeah.
Thank you both so much.
Fine.
Sure. Which method of clustering would you recommend when you want to discover how many classes you have in a segmented population?
Well, do you want to?
Yeah, so I think maybe that's kind of which method of clustering. So with phenoglyphs, I can tell you that the algorithm that does that is just k-means plus plus, but that wasn't chosen maybe because we thought it was the best method for really doing the clustering. That's more of it facilitates the workflow that we designed. But kind of like I just mentioned, TISNE is fine. I know that there's reasons why UMAP is kind of supposed to be a better method, but yeah.
Thank you so much. We have time for one more question. And again, thank you, audience, for all these great questions coming in. How many, as well as what analysis is done to show correctedness?
Yeah, this is related to the one of the previous question we just answered. In terms of evaluating the correctness, we have two ways: visual check and also qualitative adjustment, and also quantitative evaluations. So qualitative, in terms of the quality, we have biologists working with us closely. They use their biological knowledge to tell us whether the segmentation is correct or not, whether it makes sense. And we also have quantitative metrics that evaluate how close the segmentation result is compared to the ground truth.
Maybe something that would be helpful, just to follow up on that. I think with segmentation, I think one of the things that no one says, but it probably should be said, is there really is no correct answer, right? Where's the edge of a cell? I mean, this is, it kind of is, how much does it matter? So maybe instead of, if you think around an application, information quality, have some known assay endpoint and evaluate it against that, That's kind of what matters, as opposed to pixel by accuracy, usually for most of the things we care about.
Dr. Fuhui Long, thank you again for your presentation today. And thank you, Will, for joining us for the Q&A. And I want to thank our audience for their outstanding questions. As a final reminder, any questions that were not submitted or submitted after today's presentation will be addressed by our speaker via the address email that you provided at your registration. And this presentation will be available for on-demand viewing in the SBI2 virtual conference for 12 months. So please remember to share it with your colleagues who may have missed today's topic. And don't miss out on the other presentations on our agenda today. Thank you again for your participation. Until next time, stay safe, stay healthy. Take care. Bye-bye.
Thank you. Bye-bye.